Given the vast prevalence of obesity and type 2 diabetes related mortality in the United States, it is my opinion that research should focus on development of an effective strategy designed with the intent to mitigate the adverse health outcomes associated with type 2 diabetes. The overarching goal of my dissertation work was to address therapeutic strategies designed to improve vascular insulin sensitivity in vivo or within a human model. Vascular insulin signaling and its consequent vasodilatory effects facilitate delivery of glucose and insulin to skeletal muscle, which contributes to glucose uptake and homeostasis. However, with patients with type 2 diabetes exhibit diminished insulin-stimulated skeletal muscle hyperemia, which consequently limits nutrient delivery and is thought to contribute to the dysregulation of blood sugar, or what we refer to as glycemia. Loss of vascular insulin signaling, or vascular insulin resistance is what we refer to it as, results in an increased susceptibility of those large arteries or those large blood vessels to the development of atherosclerosis, as well as a decreased ability of downstream microvessels, those little tiny blood vessels in your skeletal muscle or in your muscle, to deliver insulin and glucose for metabolism and storage, or to clear that glucose from the blood in order to really control blood sugar. Type 2 diabetics are not able to really tightly control blood sugar, and we think it is because of the vascular insulin resistance displayed. So therefore, targeting the vasculature with insulin sensitizing therapies could result in improved health outcomes for individuals with type 2 diabetes. And the overarching hypothesis of my dissertation work was focused on interrogation of vascular insulin resistance, with specific emphasis placed on interrogating therapeutic interventions to improve vascular function in response to insulin. And more specifically, I hypothesized that previous exposure to shear stress, which is a force that blood flow exerts on the vascular endothelium or on the inner lining of those blood vessels, in the absence of muscle contraction, so you wouldn't even need to kind of exercise in order to improve blood flow, increase shear stress, that these forces would render the vasculature more responsive to insulin and thus make them more functional in response to insulin. And further, I also hypothesized that individuals with type 2 diabetes who displayed decreased insulin-stimulated leg vascular conductance would also exhibit reduced capillary perfusion in skeletal muscle, and that these indices would also correlate with impaired glucose disposal or reduced insulin sensitivity, reduced function. And finally, I hypothesized that participation in an 8-week exercise program characterized by increased walking would improve the microvascular insulin resistance that I've been referring to earlier, observed in subjects with type 2 diabetes. Okay, so upon interrogation of these hypotheses, I provided evidence to support my hypothesis from that first aim that prior exposure to shear stress primes the vasculature to be more insulin sensitive. And this conclusion is supported by translational evidence in vitro and cultured endothelial cells, in data from isolated resistance arteries from swine, and also with evidence from human models or human subjects. And so when examined together, these findings build a strong case to support shear stress as a primary mechanism by which the vasculature may become more insulin responsive or healthier, more insulin sensitive. Further, I also provide evidence in healthy and type 2 diabetic subjects addressing the second aim of my dissertation that suggests that increases in insulin-stimulated muscle capillary perfusion are in part determined by the vasodilation of upstream resistance arteries in type 2 diabetes, but not in healthy subjects, which is really, really fascinating and gives us further insight into how arteries in type 2 diabetes kind of function and deviate from how arteries in healthy subjects also function. So that gives us very important insight. And finally, and in opposition to my hypothesis in AIM-3, I provide evidence to suggest that regular structured exercise in the form of increased walking and in the absence of weight loss was not sufficient to improve microvascular insulin resistance in type 2 diabetic subjects, despite an improvement in the function of the popliteal artery, or that big blood vessel right behind your knee. So we were partially successful in improving vascular insulin function in that large artery, but we were not altogether successful at improving microvascular insulin resistance downstream in that skeletal muscle or in that thigh. Okay, so taken together, my findings indicate that a relatively short-term, 5 days per week walking intervention was not sufficient to address microvascular insulin resistance in type 2 diabetes. However, I also provide data demonstrating for the first time that shear stress in the absence of muscle contraction primes the vasculature to be more responsive to insulin, which is a finding that presents an intriguing alternative to exercise as a therapy. Because I achieved the increase in shear stress via single leg heating in my experiments with those human subjects, perhaps future work should address the efficacy of whole body heat therapy with attendant increases in blood flow and shear stress as a means to improve vascular outcomes in individuals with severe type 2 diabetes. So when taken together, all of my conclusions from my dissertation work highlights the vasculature as an important therapeutic target with regards to regulation of blood glucose and improvement of comorbid disease states associated with the vascular outcomes in individuals with type 2 diabetes.